Hello, today I'm going to go over the decline of amphibians lecture. So what we're talking about is this general loss of amphibian species in recent history, the last 50 years, 40 years. And what we're looking at is we're kind of going back to the beginning a little bit, and we want to see what was a little bit of the science that led them to start seeing that this was a problem. So we'll start in the 90s. In 1995, a group of elementary middle school students were collecting basic data on frogs in Minnesota. So this group was going to a specific pond collecting specimens of frogs, really normal for groups of students to do, and they just noticed that a lot of them had deformities. They noticed that about half of the frogs that they had collected had missing or deformed legs. And you can see in this image here where you've got one extra leg coming out from the back somewhere. And then even one of these regular legs is not growing right because of it. So some deformities, or several of them just had a missing hind leg. But those incidents, about half of them, that's really high. So that kind of alerted some scientists in the local area, like in Minnesota. Now taking it a little bit further. So this is going to be different groups that we're talking about, different groups of scientists. This discovery backed up what many ecologists were starting to suspect, that amphibians were in decline all over the world. The task now was to determine why amphibians seem to be more at risk in our modern environment compared to others. Now, one thing that it doesn't come up in these studies, but that is widely understood now, is one of the things, among others, is that amphibians take in a lot of material from the environment through their skin. So it makes them very susceptible to changes in the environment. Okay. So again, now we're doing a different study. So this is actually talking about before those group of elementary kids were looking at frogs. This is in 1990. Early studies from 1990 implicated parasites as the cause of the frog deformities. So in this particular one, these scientists were looking at frogs in their environment. So it was a field experiment. They were going into the field, collecting data in the field. Scientists studied a California frog and found that all of the deformed individuals had a specific parasite living within them. So in this particular one, all the ones that were deformed had the same parasite in them. And the ones that weren't deformed did not have that parasite. So just from this study alone, it's suggesting that parasites are causing the problem. So yet another experiment. So this is going to be later on. And it's taking the findings from this one and putting them in a controlled setting. In a controlled setting, that's like a laboratory setting where you control the variables. In the field, you don't control any of the variables like temperature, weather, how much moisture is in a particular stream, those kinds of things. Okay, so let's take a look at this one. The next experiment would be a controlled laboratory experiment. In this experiment, one species of frog was placed in containers with either 0, 16, 32, or 48 parasites. One species of parasite. So what they did was they took the same species of frog, caught in the same areas, and put them in individual containers with this one type of parasite. And it turned out to be a snail, but the snail had two different life cycles. And part of its life cycle, it was parasitic to frogs. So some of the containers, the control container had 0. It didn't have any parasites in it. And then an increasing amount across the other containers. The results showed that more parasites led to more deformed frogs and low survivability. And we can see that in the graph here, where it starts off with 0 parasites. You have a high percentage of them that live, and you have a 0% of them that have deformities. So no deformities, most live. As you increase the number of parasites, the survivorship, how many survive, goes down, and the number of deformities greatly increases to the point where at 48, it's 100% of the ones that survive have deformities. Okay, so that's confirming what we found out from that previous study, the field study. And then another group of scientists wants to come along and get a little bit more information about this investigation. So they want to know, maybe do pesticides also react with frogs to cause problems when there are parasites in the area? So meaning, do parasites and pesticides together cause a bigger problem? Another group of scientists then took the study further. They tested whether pesticides increased the likelihood of deformities when parasites were present. They found that multiple factors influence frog deformity. And if you look on this chart here, what you see, it's a controlled experiment again. So we have no pesticides and we have a very fine mesh. This means this is keeping the parasites out. So this is no parasites, no pesticides, and guess what? You had no deformed frogs. If you had no pesticides, but you did allow parasites in, so this mesh would allow some parasites in, you get a 4% deformity. When you have pesticides present, but no parasites, you had no deformity. So it wasn't that the pesticides were causing deformities in this particular case. However, when you got both pesticides and parasites, both of them together, you had 29% deformity. Quite a bit more than just parasites alone. So that's suggesting that pesticides that we use on plants to kill insects were weakening the frogs and making them more susceptible to parasites. And that's what we see in environmental science all the time. Many, many things interacting. Over the last 40 years, thousands of studies have found that amphibians are endangered worldwide. Studies have found that many factors, including climate change, are contributing to the record loss of amphibian species. So I talked about amphibians taking in a lot of material from their environment, but also we're finding out that just habitat destruction has been a huge factor in loss of amphibian species. All right, thank you very much.